Afternoon, everyone. Let me welcome at another round of our Fisc Talks Professor Peter Biedermann from the University of Freiburg. Today's speech or talk will be about the role of fungi in the ecology and behavior of Prague and Ambrosia beetles. I hope that everyone who's joining us online can hear us. For those of you who join us later, please don't forget to turn your microphones off. And if you will have any questions for Professor Biedermann, you are welcome to do so via our online chat. Please enjoy today's lecture, and I will give a word to Professor Biedermann.
Okay, thank you very much for inviting me, for giving me the chance to speak here. Thanks for coming. Also, thanks to the online guests. I just saw Milos Kniszek there. Just want to say hi, Milos. Nice to see that you're also listening. So I want to talk with you about the role of fungi for bark beetles, which is often not really appreciated, I think. So this one.
Do you know you're good at remote?
Okay, now it's working. Yeah. Okay. Thank you very much. So for those of you who are not so familiar with bark beetles, so we have two groups within the bark beetles, the so-called flower breeders, which are the classical bark beetles, you can say. They are under the bark making shaped tunnel systems under the bark. Of course, for the trees, it's not so nice. And then there's this other group of bark beetles, the so-called Ambrosia beetles, in the narrow sense, which make not the tunnels under the bark, but go into the wood. And these are really obligately dependent on fungi. And among those, there are only some that are obligately dependent on fungi. And I will later also explain you why this is. And in Central Europe, we have about 100 different bark beetle species. You can say about 5 different species on every tree species that we have. And maybe you're surprised with the second statement, largely economically irrelevant. Yeah, so of these 100 species, there are probably a handful that are really economically relevant. The rest is really colonizing trees when they're already dead, and really important for degradation of the wood in the forest. Yeah, but what has changed recently is that this, I would say, 95% of deadwood colonizing species, there are some of them suddenly becoming also economically relevant. And I will also talk about this, what could be the reasons for that. So when you look at the death of a tree, so here this is kind of the time axis before a tree dies and after a tree is already dead. You have aggressive bark beetles that are colonizing the tree when it's very weak, and then killing. Maybe some of them are killing the tree, and then you have a large group of bark beetles that are just colonizing the tree after it's dead, and they co-occur with fungi in that phase. And so at the same time, when you look at these trees, you also find phytopathogenic fungi together with these aggressive bark beetles, or wood decomposing fungi with the deadwood colonizers. So actually what that means is, from the beginning, bark beetles and fungi are actually preferring the same habitats, and so they are in very close contact typically. So you can say they are in a symbiosis in a broad sense, so not necessarily being good for the bark beetles, but they are in a symbiosis with many fungi, which both range from antagonistic to neutral to mutualists. And so the question now is, but why are so many bark beetles then in a mutualism with fungi? So how do these mutualisms involve? So for mutualisms to involve in general, you need organisms to interact very commonly with each other. So they need to co-occur in time and space. this is already met. I just explained they are co-occurring with fungi in the same substrate. And then the second thing you need for a mutualism to evolve is tradable traits. So bark beetles, the fungi profit from bark beetles by dispersal, or may potentially profit by dispersal of spores. You can imagine a tree protected with bark. It's really inaccessible for fungus spores. But when a bark beetle is making a tunnel through this bark, then it's really open for the fungal spores to grow. Yeah, and some other traits, I will talk later about that. And okay, it's again not working. And beetles on the other side, they profit from fungi mostly by enzymatic capabilities. You know, fungi have a lot of potential to degrade chemicals, to produce certain chemicals, what insects cannot do. So there are traits that can be exchanged. And a third really important factor is partner fidelity. That means for an evolutionary process to start so that a mutualism can evolve, or a dependence between organisms can evolve, you need these organisms to stay together over multiple generations. And this is actually maintained in many bark beetles by so-called vertical transmission of fungi. So what, if you didn't hear this word, vertical transmission, what does it mean? It means that, so here you see the generations of an insect. And in vertical transmission, it would mean that the mother is taking the offspring that are emerging from a nest are taking the fungi that were with them in the natal nest with them to found a new nest. And so over time, if the beetle profits from the fungus, and there can be a feedback evolving between these two partners. Whereas in horizontal transmission, it's much more difficult that a feedback evolves, well, because in horizontal transmission, what it means is that the symbionts are acquired anew every generation from the environment. And so you never have the genotypes of the insects linked with the genotypes of the fungi. And then these kind of symbioses are much more unstable. A lot of the very important or the obligate mutualisms in nature involve vertical transmission. And how do these bark beetles do that? That was discovered already about 50, 60 years ago by a European and an American about the same time. Bark beetles have organs, or many bark beetles have organs within their bodies that with openings to the outside of the body, where they store spores of beneficial fungi. So you can see here, these are huge organs in some species, and within these organs, they selectively propagate spores of beneficial fungi. So here you can see this is an Ambrosia beetle where you can see the lining of fungus on the tunnel walls. And here I want to briefly explain to you how such a mycotangium works. So when a beetle is crawling around in these tunnels, so let's say after pupation, when it's crawling around in the tunnels, there are fungi growing on the tunnel walls. And then the spores are picked up by this mycotangium. And the interesting thing is when you then take out these beetles that are inside the wood, you will find a community of different fungal spores within this mycangia. And only when the beetles are starting to disperse, they activate their mycangia. So they actually produce some there are some glands that produce specific substances. We don't know yet what they are. And these substances then select just this one beneficial fungus that they want to transmit. And so you really have to catch the beetles when they leave the nest, because then there's only the one fungus that you're interested in. Or if you're interested to look what fungus is beneficial, then you should catch the beetles when they emerge from the nest. And then when so here is then the host switch, then they colonize a new host tree, and then these are really active, this mitochondria, and so that they produce a lot of spores, and then these spores are then also smeared on the tunnel walls in the new tree, and then they regrow again in the new tree. Was this understandable? Yeah, and what is really cool is that these mitochondria have so many different forms and have evolved so many times independently in this group of bark beetles. You see, these are always the white structures. So they, as I will later explain, they evolved multiple times independently. And they're really super specific. Typically, one species of beetle has one species of fungus. Yeah, and actually, this is something really new. You see, it's just a week old. It's also in our famous European spruce bark beetle. There's now a mitochondrium described since a week. It's still in preprint, but they found an oral mitochondrium. For a long time, it was hypothesized this beetle doesn't have any mitochondrium, so no obligate mutalis that it's transmitting, but actually they found one at the mouth parts of this beetle. So here these are the mandibles, and here is a small opening, and behind this opening there's a structure where they transmit Rosmania penicillata mostly. Yeah, so but this sounds now really simple, and it sounds like okay, you have one beetle, one fungus, and so it's actually quite simple to study. It isn't so simple. So when we, for example, here these are newly emerging pests here. You can say the fear bark beetles or fur bark beetles here in Central Europe, and we don't know anything about their fungal mutalis. And so we started looking at the fungal mutalists, and you see, or there's fungal symbionts, and you see there's a whole community of different fungi. If you really take the beetles that are, as I said, that are emerging from a nest, if you collect them, you still get a community of different fungi associated with these beetles. And so what we did in this study, we stored the logs before, so after collection in the forest, with the logs were colonized by bark beetles in one population, and then we stored the logs at different temperatures, and you can see this storage of the logs in different temperatures had an influence on the fungal community that emerged. So there are more of the red fungi here, and when it's warmer, and when it's stored warmer, and more of the blue when it's stored colder, and these two are possibly the important fungi, but that we cannot say just by looking at this by metabarcoding. So this was done by metabarcoding. So there you have a different community of fungi. And when we then want to find out what are potentially mutalistic fungi, we usually do tests like this one, so that we have one fungus. So we inoculate one fungus on one side of an agar plate. On the other side, we have just agar. So this is just sawdust with agar. And then we put a male or a female onto this plate and check how it's behaving. And And typically, when the fungus is interesting for them, then they stay closer to the fungus or even bore in. And that's what you see here. So these are all the fungi that we cultivated out of the beetles from the beginning, and we saw, okay, 2 of the fungi are actually really preferred by the beetles, so they start to bore in when the fungus is colonizing the media. And one fungus was actually repellent. So, and some of the others, it's unclear. So that's how we can find out if a fungus is good for them. So let's go back to the tradable traits that I said at the beginning. So what benefits can fungi have for the beetles? So for that, it's good to think about plant tissue and how valuable it is as a food source for insects. It's actually, so we often think plant tissues are really good for herbivorous insects, but actually plant tissues are really biased in their elemental composition. So there's a lot of sugars, but very little nitrogen, very little phosphorus, very little other micronutrients. And also, typically, plant tissues are also full with toxic compounds. So, and if we look requirement, what the beetle requires to build up within its body is, for example, here the composition of nitrogen in the beetle compared to food or xylem and phloem. You see that the beetle with its diet has to actually accumulate a lot of this nitrogen, and even more strongly for phosphorus. It really either needs to feed a lot on a lot of plant tissue, or it needs some help from microorganisms to acquire these nutrients. So former PhD student of mine, Max Lehenberger, and also some other people, they looked at that, and Max looked at that in these fur bug beetles that I explained before, with these 2 fungi that we showed to be beneficial. And here you see, this is the total amount of toxic substances, phenolics and terpenoids within fur phloem. And after the fur phloem is colonized by the Geosmitia fungus and by the Ophiostoma fungus. And you see, it's very strongly reduced, all these toxic compounds. And that's probably one of the reasons why the beetle like to dig into this kind of phloem when the fungus has colonized it, because the fungus has actually to some degree detoxified this substrate. But not only detoxification is important, but also degradation of polymers and actually additional nutrition. So this noise is really annoying.
I wonder if you can just turn all the volume off. Here it comes.
Yeah.
On the computer. There. Or could you just take the whole computer volume down?
It's not the computer.
I know, but it's coming through the computer.
Sorry. Okay. Yeah. So here he also looked at the total amount of sugars, and you see also this single sugars, and you see that also the fungi are apparently degrading cellulose or hemicelluloses, because then there's also more sugars, monosaccharides present within the phloem when it's colonized by fungi. And also the amount of amino acids is also much higher than in the phloem alone. So yeah, the fungi are apparently making it better as making the phloem better to feed on. Yeah, and something interesting has been discovered also in the US with Dendroctonus brevicomes a few years ago, because they have shown that actually some of these elements, the nitrogen, phosphorus, but also other elements, are not only brought or not only coming directly from the phloem, but actually the fungi growing into the xylem quite a bit, and then catching and relocating the nitrogen and the phosphorus for the beetles into the phloem, where the beetles are then feeding. And this has been done with bark beetles. We also thought, or Marx thought, oh, it would be quite interesting to also test this with Ambrosia beetles. So with the beetles that are really breeding within the wood, within the xylem, which has even less nutrients. And so he tested this with 3 different Ambrosia beetle species. And what he did is he looked at the elemental composition 5 centimeters away from the tunnel, close to the tunnels, and then within the tunnel systems, the fungal layers, they looked at the elemental composition. And here, this is a visual representation of the concentration of different nutrients. You see always on the left side. So here, the left side, this is the tunnel system with the fungal layers, and on the right side you have the xylem. And you always see within the tunnel system, the concentration of different nutrients is much higher than outside of the tunnel system. And if you really quantify that, what he found is 5 centimeters away from the tunnel system, phosphorus concentration is very, very low. 1-2 millimeters next to the tunnel systems, it's a bit higher. And within the tunnel system, it's really up concentrated 50 times. And that's actually, if you remember the values before, with this, the beetle needs 50 times, needs to actually accumulate 50 times or concentrate the nitrogen 50 times and phosphorus 50 times. It's the same number. So actually, when the beetle is just feeding on the fungus, it's about the same concentration of nutrient it needs for building its body. So yeah, apparently these Ambrosia fungi also translocating the nutrients towards the nest. Okay, that's about nutrition. And there's also, this is already known for quite a while, so since the 80s, that some of these fungi that are associated with the beetles are also detoxifying, not only detoxifying, but also causing tree virulence. Like these fungi to the trees, you then see lesion zones that are different sizes. So here you see these are different fungi that are associated with the European spruce bark beetle. And these are the lesion zones. So some species of fungi are really causing big lesion zones. And the lesion zone is not always a proxy for the virulence, but very often. In the Ipsilographos, this fungus, Endoconidia4 polonica, is actually the most virulent, even though the virulent zone is not so strong. And it has been quantified that these Ipsilographos beetles, these are really aggressive bark beetles that can kill healthy trees, but only above a certain number. And what is really interesting is if these beetles are having Endoconidia4 polonica with them, then they only need half the number to kill a tree, because the other half is then because of the virulence of this fungus. And this is really important for the beetles, because when they colonize a tree in masses, then the competition between each other is also huge. And so if you are only, but if you can colonize a tree in half the amount of half the number of beetles, this means the competition is much lower than between the offspring. Okay, so I think it's quite clear that the beetles profit from the fungi, but do the fungi also profit from the beetles? Yeah, so I mean, probably the first, the most important, most profitable for the fungi is the dispersal of the spores to inaccessible substrate. But then also the beetles are weakening the tree by tunneling, by disrupting the water flow, which makes it also better for the fungi to colonize the substrate. And what has been hardly appreciated also is that the beetles actually also have a chance to manage the fungal communities within the nest. So this is a video from an Ambrosia beetle that was reared in artificial media. You see here, this is the mother beetle that was just crawling to the larvae. And so have a look at the larvae and the beetles and try to find out what they are doing. And in a few seconds, I will then ask you what type of behaviors you think you saw. So these are young beetles. These are daughters of the mother. The white layer here is really the fungus layer that is grazed off by the beetles. But what behaviors do you think did you see?
Feeding?
Weeding, yes, weeding. So this glass here is super clean, although there are lots of microbes growing in there. So they're all the time clean. I mean, glass is artificial for them in their environment, but you can see, wow, if I would remove the beetles within a few hours, this will be covered with fungus. So this and so right now it's super clean. And what else did you see? Did you see some interactions between?
Was the young beetle grooming the larva?
Yeah, exactly. The young beetle was grooming the larva, and before that there was also larvae grooming each other and larvae grooming pupae. So actually the most common behavior in the nest is actually allogrooming, which is when we keep beetles, so we did some experiments, when we keep beetles hard in the tunnels, so if we cut them off from the rest of the group, then they will quickly overgrow with fungi and many of them will die actually. So they have to groom each other. But this grooming can be also selfish in a way because you're potentially feeding on these fungal spores. And then there's also nest hygiene. At the beginning of this video, I don't know if you saw, the mother was actually taking some sawdust from the larvae that was pushed from the larvae to the mother. So the mother they're together cleaning the nest from feces. So you see that there was hardly, so this is feces here, but generally it's pretty clean in there. So, and they pass on this feces to the outside. Yeah, and we are not sure if is there really fungus tending, or are they just grazing on the fungus, and by this grazing they are changing the community. Anyway, what we discovered is that there's division of labor in this Ambrosia beetle nest actually. So these are really social beetles where the larvae do different tasks than the adults, and yeah, and both is really important for keeping the stability of the nest. And there's actually one a new social Ambrosia beetle discovered in Australia. We don't know much about its behavior. It's living in living trees. We cannot rear it in the lab, so we don't know how it's behaving within the nest, but we know that there are adult daughters that stay back with the mother and never reproduce. So like really, you have a queen beetle and workers that stay back with the mother. Yeah, and this is not only, so the most social beetles are the Ambrosia beetles that are really obligately dependent on the fungus and that are breeding within the xylem. But also among bark beetles, you can see a lot of things that are similar. We also see that the bark beetles are cleaning their tunnels. They are smearing some secretions. We are not sure yet what they're doing with that. They're cleaning their eggs. They're putting. So in Ipterographus, it was discovered last year that they actually cover their eggs with pieces of sawdust that are full with specific bacteria, so that at the beginning, when the larvae hatches, it's just feeding on this starter culture in a way to get its first gut microbiome from the provided by the mother. Yeah. So there's social microbial management, and what one of my PhD students did some years ago, she actually let a mother start a nest, and after a week, after starting the fungus community, but before laying eggs, she removed the mother again, and then after, I think it was 30 days, she looked at the fungus community with beetles and without beetles. What she saw is that without beetles, there are more fungal pathogens in the nest, and the two food fungi that are really important for the beetles are found in lower abundances. So apparently, this beetle activity can promote their nutritional fungi. We don't know yet. Is it just the feeding on these fungi, like in cows that are grazing on the pastures, that they are by just feeding, they are selecting a certain community? This could be the case, or do they really propagate certain antibiotic bacteria or selective substances? We don't know yet. Okay, so if we look now in history, how did this all start? So how did this symbiosis or this mutualism start? It started with the symbiosis, so that there were just fungi occurring at the same time with the beetles in the tunnels, and these fungi probably profited from being dispersed by the beetles. And then at some point, some fungi turned out to be a bit beneficial for the beetles. So when they were present in the nest, the beetle had a bit more offspring, and so the beetle actually then tried to bring this fungus with it. And then at some point it developed the first organs to, first maybe it was just sticked, but then the beetles evolved the organs to transmit the fungus. Yeah, and at some point this fungal association also allowed the beetles to go in the much less nutritious xylem, where they cannot live without fungi. Yeah, and this fungal mutualism is a super successful strategy in bark beetles. So this is the whole phylogeny of bark beetles. In black are all associations that are not obligate with fungi, and in blue you have the obligate associations, and you see 16 origins, independent origins of obligate fungus feeding in beetles. And if you look what kind of fungi do they have, it's interesting. They have also 10 independent origins in the fungi, so they're really cultivating different kinds of fungi. And this may be also explaining why the mecangial structures look so different, because they independently evolved for different kinds of fungi. And if you compare that with other groups of insects, actually, you have these fungus or obligate fungus associations is something not so common in insects. You have just 33 origins in total. There's the leafcutter ants, for example, maybe you've heard of them, or fungus growing termites, or wood wasps. These are famous examples. But half of all the origins are actually in this group of beetles. And also in the fungi, it's about the same. It's most of the origins of independent origin fungus mutualism are in this group. Yeah, and then you wonder, okay, why are there still some bug? If it's so profitable for them to have fungi, why there are still so many bug beetles that didn't evolve obligate mutualism? And if you look at the occurrence, for example, of bark beetles that are obligately associated with fungi, you find them mostly in the tropics, and the further you go north, the less you find them. And so I think it could have something to do with being too cold for the fungi. And what you also see is when you have a tree, these bark beetles that are colonizing the stem, they often are associated with fungi, but then if you go out into the twigs, then it's probably too dry for the fungi to grow. And then the beetles are just feeding on or maybe it's also more nutritional, the twigs, I don't know. But these are two patterns. Additionally, sometimes fungi may not be necessary if beetles are really colonizing really old wood that is already colonized with other fungi. They don't need to bring their own fungi. And what is also the case is that in some cases when beetles are really crowded, then it's typically colonizing trees in very crowded conditions, then there's a lot of horizontal transmission of the fungi, and so they cannot I think they cannot develop an obligate association. Okay, so are you still up for some more slides about what is happening with bark beetles in the Anthropocene? So yeah, we are talking a lot about bark beetles now because they're causing quite some damage, especially in temperate regions. And so there you can observe two kinds of phenomena. The first phenomena is that the damage by aggressive bark beetles is massively increasing. It's especially here in Central Europe. It's the European spruce bark beetle, Ixtabographus, that is having unprecedented outbreaks here, especially in the Czech Republic some years ago, but also in other parts of Europe. And the other phenomenon we observe is that typically harmless bark beetles, or that in the past were harmless, are becoming aggressive now. So let's have a first look at the first phenomenon, the damage of aggressive bark beetles. What a lot of people, what isn't clear for a lot of foresters actually is that aggressive bark beetles are not per se killing trees. So they are not dependent on killing trees. They're actually adapted to freshly fallen over trees. So especially our European spruce bark beetle, this is what this beetle prefers. It prefers wind-thrown or snow-broken spruce trees. And in this wood, it can flourish, or it does really best. So, and here you see the population growth let's say in a healthy forest. So you have some dead trees, and the beetles can just the more beetles there are, the less the population grows. But what is really strange is that you have this kind of curve. Can somebody explain to me what you think, or why what is happening there. So apparently at some beetle abundance, you have another peak of growth, or the population can grow again. What happens at this transition? Do you know what can you imagine?
I mean, I guess they're overcoming host defenses. Probably.
Exactly, yeah. So this is when they can kill healthy trees. So the threshold, they can only colonize this kind of wood, but above a certain threshold, they can kill trees, and they can do this with by doing mass aggregations, by following other beetles that are already made a hole into the tree and producing some pheromones. When they are around at this population size, you can see that some individual trees are attacked by a lot of beetles, and they do this with aggregation pheromones and by the help of plant pathogenic symbionts. So when plant pathogenic symbionts are present in the population, the beetles are doing better. And what is now happening with climate change actually is that the tree health is suffering. So we get reduced defenses, and so the beetles need less. The population needs to be less high for the beetles to overcome these trees. And what happens if there's less competition within the tree is unfortunately also that the beetles are producing better within these trees because they are not competing with so many others. And this is kind of a deadly feedback, because then you also have more beetles within the forest and reduce defenses because they're reproducing better. This kind of trees, and that's why we see these huge outbreaks also only in these phases with droughts. So, example, here in Europe we had this 2017, 18, 19. It was really dry years, and then we had these huge outbreaks. Yeah, and about the other phenomenon, harmless beetles become more aggressive. And I think there's something similar is happening actually. So one example for harmless beetles are fur bark beetles that in the past very rarely were killing trees. And so this is the growth curve that I drew here. So in only very rare cases, when the tree when they were really in very high populations, they were able to kill trees. And what's happening now is that, again, the same thing like in Ipstogographus, that we have a reduced tree health, and then suddenly these beetles are able to also kill these trees if they are in high enough numbers. The first option. But there are also other bark beetles where this is not true. For example, the elm bark beetles. You all heard of the Dutch elm disease. And the reason why these bark beetles are able to kill trees that they couldn't kill in the past is actually phytopathogenic fungi. And in this case, really invasive phytopathogenic fungi. And suddenly so in the past, the beetles were just colonizing dead trees, and now they can actually colonize living trees, and their fungi will kill the trees. And the third option, what we can observe in the Mediterranean now also, is that we have new invasive bark beetle species that are actually harmless in their native range, but they kill trees in their invasive range. So these are, and very often people are not looking closely what actually is the cause for this. So there are different causes for the beetles to become more aggressive. It can be climate change, can be new invasive fungi or the beetles are themselves invasive. Okay, how to move forward? So I think it's quite meaningful to really study the bark beetle ecosystem more closely. So what organisms occur within these nests. So I've explained to you there are some fungal symbionts in this nest. There are also bacteria. We also started to study some of these bacteria in these nests. And there are also many natural enemies within these tunnels which have not been really explored much. So there are mites, there are endopathogenic fungi, there are nematodes, parasitoid predators, and actually viruses also have not been studied in bark beetles, although they are known in some other insects to be really super important for regulating population density. Yeah, and so things that we are really interested to study in the future, and maybe some of you are also interested, and then we can also collaborate, is can fungal symbionts of the bark beetles affect population dynamics? Can other natural enemies be used for management? Can the mutualisms between the bark beetles and their fungi be disrupted? Maybe so in management, we often think about, okay, what can we do against the beetle? But we are not thinking that there are organisms the beetle needs. And so maybe we can also look more closely at the fungal parasites of the beneficial fungi. Then the basic question is, how do mycotentia work? And also, can we use fungal volatiles for attracting or repelling bark beetles? And Anna actually here is also doing some studies about this. Okay, with that, I want to acknowledge my group, because most of these things hardly anything is done by myself of these things. And I want to point out a new master program we have in Freiburg. Maybe you have some bachelor students or so that would be interested in that. It's called Forest Master Program in Forest Ecology Research. And what we try, it's existing now since 2 years. It's now going in the 3rd year. And we want to train students as modern forest scientists. So we want to train them how to understand the interactions among the plants, animals, microorganisms, and also abiotic factors. So in the center are the trees in the forest, in some cases also the stands, and we want to understand how the biotic factors and the abiotic factors that influence these trees. And the students will get some training in modern methods, genetic methods, chemical methods, microbiological methods to really study this in depth. And yeah, so this is the program. It's actually, I think, quite unique in Europe, because mostly in forest sciences we have studies that are focused, or at least half, on economy, and in this program you would be really focusing on research. So this is targeting for research. So if for students that are interested in research careers within forests, so that you become a researcher in the research forest scientist, actually. With that, I'm at the end of my talk, and thank you for your attention.